All right, good morning, everyone. My name is Dr. Prentice Williams, and it is my pleasure to introduce our speaker for the ECE colloquium today. Our speaker today is Dr. Megan Matthews. Megan holds a BS, MS, and PhD from NC State. She actually graduated from this department, where she developed, as part of her PhD, a multiscale model of lignin biosynthesis in poplar trees. Megan went to the University of Illinois, Urbana-Champaign, after graduating here as a postdoctoral researcher to develop multiscale models, crop models for the ripe and crops in silico projects. A year after she actually went for her postdoc, she joined the faculty in the Civil and Environmental Engineering Department as an assistant professor in January 2021. There, her research focuses on developing multi-scale plant models that integrate information across multiple levels of biological organization, and using those models to, one, explore the impact of changing environments on plants, and two, identify engineering strategies for improving plant development and growth. Again, it is my honor to introduce to you Dr. Megan Matthews, especially because she actually came out of our research lab. So please welcome Dr. Matthews.
Hi. Thanks for the introduction. I'm excited to be back at NC State again. So thank you all for coming to my talk to listen. As you heard multiple times in the introduction, I'm going to be talking some about multi-scale modeling of biological systems, specifically towards the efforts of how we can hopefully use them towards engineering plants. And so we use math to model a bunch of different types of systems. In civil engineering, some of my colleagues use it to look at transportation networks, transportation flow. Here in ECE, things related to power systems, solar network, power grid, or electrical circuits, all different ways that you all have used math at different kind of levels or scales to describe and work with these types of systems. Can use the same type of math and analysis approaches when modeling biological systems, including in plants like crops. And so the way this kind of works, the same way that we model the flow of how voltage changes in a circuit using ordinary differential equations, we can do a similar approach with different parts of biological systems, like metabolic pathways, like photosynthesis, and write rate equations to describe the flow of information in these pathways using differential equations. And so the goal of kind of developing these models, of exploring or using computational tools, math, to describe these biological systems, is that we can test a lot of different scenarios in silico or through these simulations than we could test in the field. And so we can develop these models, maybe learn a little bit of different things, and kind of just explore of, what if I make this change to the model or the system? If I could do that experimentally, what type of output or change in the plant or crop would we expect to see? You can run a whole series of these types of tests and simulations and hopefully identify promising strategies or ways of changing or engineering the system or plant to meet goals that you're interested in. Then you can take the most promising predictive strategies and test them out in the field and experimentally. The goal is then to take the results of what happens in the field and then use that to hopefully model it well. Maybe some areas of improvement, no model is going to be perfect. And so use the data, what we learned then, to kind of reevaluate, update, improve the model, and kind of have this cyclical kind of predict, test, learn cycle with the data. Some of the questions around doing this, especially are of interest in kind of a lot of the global challenges facing the community, things related to food security, energy security, environmental sustainability. And so, let's see, go over here. What we have, or I can even do it on here, it's probably better. What we have for the, there we go. In this picture, we have 4 different crops here: corn, rice, wheat, soybean, which are 4 of the major crops that are globally important, especially on for food. and is showing kind of historically the rates of increases of the yield in the crop that we've seen. There's been kind of a linear trend of increases. As going out into the future, though, if we kept on this similar trend of increase, this shows the solid line what is expected, we would expect to see if we kept on the similar rate. However, to what's expected that we're going to need in order to meet the global food demand and energy demands is what's shown in the dashed line. And so there's this disconnect of if we continue on improving yields and crops at the rate we have, we're not going to meet that demand. In addition, at the same time, we are going to have different climate conditions as we go into the future that are going to impact how these crops are grown, where crops can be grown, and that is also going to impact the expected yields and how these plants are going to adapt or acclimate to these different conditions and what we can do with that. A lot of these previous increases in the yield I've seen before have mostly been due to changes in like fertilizer management, land use change, among others, which can sometimes lead to other environmental consequences, and in the future, wanting to move towards improving the yields, but in a more environmentally sustainable way. And so plants have complex regulatory strategies in order to deal with their environment. They can't get up and move away when the environment changes, and so they need to adapt and acclimate, and they do this at a couple different levels of biological organization. Kind of shown here, we have the genes, which are essentially the blueprint of what happens in the plant, going down to kind of the RNA, proteins, metabolism. It also has different scales of spatially and temporally of how things change as plants grow, and also the communication between different parts of the plant. And then, of course, there's also going to be the inputs and kind of outputs of how these plants and crops interact with the larger environmental systems, including the weather inputs, and also if we are changing yields and crops, do we expect that to change kind of the larger ecosystems that they're in. And so that brings me to kind of the two projects that I'm going to talk about today. The first one is related to improving wood traits for conversion to biofuels. And then the second one will be talking about engineering crops for the future. So this first one is looking at particularly working in poplar trees with collaborators here at NC State in the Forest Biotechnology group. And so cellulose is part of cell walls, specifically the secondary cell walls of plants, and it's the most abundant organic compound on Earth. And this is what is used a lot for like the biofuel, the bioethanol production that you may hear about is the cellulose, also used a lot in the biomaterials industries. And so getting this cellulose is of interest and important for a lot of different industries. A barrier to extracting the cellulose is the lignin, which is also in these secondary cell walls, and it's kind of entangled with the cellulose. And so the lignin makes it difficult to extract the cellulose from these cell walls and adds a significant kind of cost to removing the lignin in order to get the cellulose, which is why you don't see as much of the bioethanol in kind of when you go to the gas station as people were, I think, hoping we would be at this point. It's because the cost point of extracting the ethanol and creating ethanol is not there, and a large part of that is due to this lignin removal. But lignin does have an important role in plants. It helps give them their structure. It helps to conduct water throughout the plant, and it also acts as a barrier to pests and pathogens. So you don't just want to completely remove lignin from the system. And so the question is, can we engineer trees to have more desirable lignin traits to make that extraction of the cellulose easier without harming the physiology or adaptation of the tree itself? And to do this, we started with what's shown here is a metabolic pathway of the lignin pathway in plant cell walls. And so this shows the metabolites in the circles, where a metabolite is things like, if you think of the sugar, the sucrose, glucose is a type of metabolite. Another type is a amino acid, which is, if you are into like nutrition and fitness, you may care about the types of amino acids you're eating and getting in your diet. The first metabolite in this pathway is phenylalanine, which is a type of amino acid. And then these arrows, and what's in blue, are the enzymes, which are a type of protein that bind to the metabolite, alter it in a little bit way, and then release a slightly different metabolite. And so you go through these kind of steps in this metabolic pathway. You're starting with, in lignin case, phenylalanine, and you end up with going through here. This is the typical in poplar trees, at least kind of typical flow of information through this pathway, and end up with these three main outputs of the pathway. We call these the monolignals, include the H, G, and S subunits. And so we can make changes to the flow of information in this pathway through different modifications to the pathway, such as completely knocking out or removing one of these enzymes such that we now can't have the flow of information through. Ways of doing, or sorry, I'll get to that, which would then change kind of this flow of information through changing our ratio of our outputs, the type of lignin, and changes to the phenotype, which means the physical traits, so how easy it's to extract, the cellulose, or things related to like the density of the wood in the tree. Those types of traits. And so this has been shown, this has been done experimentally, but it's been difficult to really understand or tell just by looking at the metabolic pathway or models of the metabolic pathway of how to really make changes here to get the desired output wood traits that we want. And part of that reason is because our model is just looking at this kind of metabolic layer right now. But when we're making changes to how the information flows through the pathway, what we're really changing is these RNA or the transcript amounts. And so the kind of flow of information here is you have your gene, which is the essentially kind of like a cookbook, making copies of specific recipes, which would be the RNA, get passed out, that get translated into these proteins, which some of which are these enzymes here. And so if we are making changes here to the RNA or the amount of copies of the recipe we have, we are changing the number of the enzymes available to kind of catalyze these reactions in the pathway. But there are other levels of regulation going on that are occurring that were not previously captured if we just change the amount of the one enzyme that we're targeting. Which is why we then decided to create this kind of multi-scale model that captures all of these different kind of layers of biological organization to capture the cascaded impact of the modifications going through. And this model is composed, that we developed, is composed of kind of three main parts. One is looking at these relationships between the RNA and the enzymes in the pathway of can we predict how all of those enzyme abundances may change when we are making changes to the RNA? This information is then fed into a mechanistic model of that metabolic pathway. And then we took the outputs of this to then try and do some phenotype prediction. So mostly on this part of my talk, just going to focus on this first part and a little bit on the second part, and then cover the model as a whole. And so for this first part of this model, the initial version that we started with made the simplifying assumption that each protein was kind of directly and only related to its associated RNA or transcript. And this makes sense. The protein is directly translated from the RNA, and so there's going to be that kind of one-to-one relationship. And we can see that for some of these enzymes and transcripts, that worked out very well. There's this very strong linear correlation. But for other ones, it's not so much. It suggests that there is some other types of influences, indirect influences, that when we are targeting other genes in the pathway are also impacting the amount of protein that we're getting here. And so our goal then was to develop a model using the data and information that we had in order to capture some of these indirect influences between the RNA and proteins. And we did this using a more data-driven approach based around linear regressions, but defined each transcript or protein in the system essentially as a linear combination of the other transcripts or proteins in the system, put all of those together into one matrix or system, shown here, matrix B, and then used a sparse maximum likelihood approach to solve for our weights in this B matrix, shown here. And so the results of this are shown in this heat map here, where the green boxes show a positive indirect influence, the red boxes are a negative or inhibitory indirect influence, and the y-axis and x-axis are the same, and so this just has all of the RNA or transcripts, and then all of the proteins, and so each quadrant is showing essentially the relationship from either transcript to transcript, protein to transcript, protein to protein, or transcript to protein. And we see here that we have that one-to-one relationship still captured, which is good, because we know that that exists, between each transcript and its protein. And so the goal, though, from this is not just to kind of create or determine what some of these indirect influences are, but it's really to use this as a more predictive model such that we could, if we wanted to make changes to some of the, to one or multiple of these RNA or transcript abundances, we want to be able to kind of predict the protein outputs for all of the proteins in our model, which is about 20. And so we were able to do this with the type of model that we developed. And just to show a couple of examples, comparing it to our original version where we don't have any of these indirect influences, I'll show the outputs from one, which did particularly well, in my opinion. But here, so we're looking at if we want to decrease the amounts of these enzymes, the CALD5H1 and 2. So there's two of them, but they're the same enzyme family, and they catalyze these two reactions shown here in the metabolic pathway. This plot on the left is showing our wild type amounts of those transcripts or RNA. And so the wild type is our control condition. And then we have these three experimental that knock down to differing levels the amount of these transcripts shown here. And so the amounts here were the inputs to both versions of the model that we're showing, and we're wanting to predict, now we predicted all of the proteins or enzymes amount shown in the pathway. I'll show you for the HCT6 protein, which is up here earlier in the pathway. And we see for the transcript, the experimental results are in this gray bar here, shown here. The new model that has the indirect influences is the red, and the old model that just has the one-to-one relationships is yellow. And here, experimentally, don't see any change from wild type in the transcript amounts. And both models capture that. So with, in the old case, everything else, we're not targeting knocking down the HCT6, so that's just assumed it's going to stay wild type anyways. In this case, that happens to be true. Our model, we don't make that assumption, but we're still, or the new version with the indirect influences, we don't make that assumption, but we're still predicting that. When we look at the proteins, however, we see experimentally measured an increase in the HTT protein. And because with the older version of the model, it's just assumed to stay at that control, that wild type level, we don't capture that influence. But with our new version, we do predict this higher amount of the HCT6 protein. And this is important because this abundance of the protein is what is actually fed into the metabolic model. And so capturing those indirect influences or the untargeted changes to those values are important for capturing the cascaded impact of the full model. So then, like I said, those protein values get input into our metabolic model here, mechanistic, and then we run this model to reach a steady state of the fluxes or those reaction rates in the pathway, and then those outputs are used in this phenotype prediction. And so there are different levels of information known about kind of each of these steps. The metabolic model, we know this pathway fairly well. We're able to do a really mechanistic model of this. With the transcript protein model that I talked about, we know a little bit, but it was still more of a data-driven approach for modeling. With the phenotypic prediction, it's still really not understood biologically how we go from those monolignals into the full polymerized chain of the lignin that's entangled with the cellulose. So it's still very much a black box to biologists, really, how that process is working. And so we again took a more machine learning type approach or data-driven approach for the phenotypic prediction. In this case, I used random forests, which are popular in biological applications because they are fairly robust. You don't need as much data to train them as you do with some of the other machine learning or deep learning techniques, which is a strong barrier in kind of biological applications, is the amount of data we have. But they still are able to capture these complex relationships between pathways. So it can capture nonlinearities and those types of relationships as well. And so trained the model, the random forest model, machine learning approach, and then looking at now kind of this cascaded impact of the full model, again, comparing the when we're incorporating these indirect influences on the transcript protein model versus not incorporating running through, we can again compare some of our results to show how well it's doing at the phenotype prediction when you compare those two transcript protein models. And so again, looking at this CALD5H1 and 2 knockdown, we can look at, these are some of the selected traits from this full now multi-scale model that we're looking at with our using the new updated version transcript protein model in black, the old version with just the one-to-one relationships in the gray, and experimental measurements are the red dots. And so we have some specific kind of lignin traits, including the ratio of the amount of S subunits versus those G subunits, as well as some of these other linkages that are involved in the full polymerization of lignin, such as beta-5 is one of them. And then we also looked at some traits that are not as directly only related to lignin. Things like looking at the amount of the glucose or xylose that we could, which is that part of the cellulose that we want to extract from these trees. In this case, looking at unpretreated. And showing here, key message from here is showing that basically the black lines do a better job of fitting that data when we include those indirect influences below. Kind of the one exception here is the H subunits, which didn't do as well. The large reason for that was the range of data we had for the H subunits when we were training the model was not very large and had some few issues with it. So kind of explains that. Okay. So then the goal of this is to use this full model now to make predictions of to identify how making changes at the amounts of RNAs for different genes, how is that going to change our predicted lignin and wood traits? And so an example here show the combination of decreasing now these, it's called the coenzyme T family of RNA or enzymes, which are these reactions, as well as the POW reactions, looking at decreasing the amount of these transcripts in the system and seeing how it impacts. In this case, we're looking at the lignin content. So at wild type, you have wild type's amount of lignin, and as you start to decrease these, you get a decrease. But you have to decrease a fair amount, especially in the case of the POW, before you start seeing the decrease in lignin content here. But there's some combined impacts from decreasing both at the same time. And so you can use this model, and it has been used recently to kind of explore and identify different regions and ways to identify specific gene or gene families to target to decrease or increase in order to get specific traits that you want, such that you can look at, we want to keep the density maybe at wild type levels. We don't really want to impact the tree health too much. So what changes can we make that would keep density at wild type levels, but increase the amount of sucrose or xylose that we're getting from the cellulose, our ability to extract that information. And so a lot of that work has been going on to kind of validate, and that experimental work using the model predictions has been going on the last couple of years. But to wrap up this portion of the talk, this is basically combination data-driven mechanistic approaches based on the amount of prior knowledge we know of the system, and that capturing the effects of the cross-regulatory influences at the transcript and protein levels was important and allows us to capture the cascaded impact that we see now at the physical traits that we really care about better than just looking at or making the assumption of the one-to-one relationships. And can use this model now to explore and identify promising engineering strategies to look at increasing our ability to extract the cellulose by impacting the lignin in the trees. Okay. So that brings me to the second part of my talk, which is now looking at engineering crops for the future. And so let's go back to the slide that I had at the beginning. Again, so the lignin work is a lot on the kind of bioenergy side of this. The project I'm talking about now is looking more on the food security, environmental sustainability aspects of these global challenges, and wanting to increase the yields of crops to meet these future food demands, as well as deal with future climates and how we expect to do with that. And so the question is, can we engineer crops that are more efficient, which could include things like looking at higher yield or decrease in the amount of needed land and water resources? And so to do this, now the lignin model was looking more at kind of the molecular level, and this work is more at the kind of crop and crop physiology level, along with its connections with the larger ecosystem, environmental inputs to the model. And so this model that I've been working with is called BioCrow, scales from the biochemistry of photosynthesis to whole crop growth and yield, and it takes in the inputs of the atmospheric CO2, as well as water and some nutrients and the solar radiation expected by the plants. And it's able to capture photosynthetic responses to changes in temperature and atmospheric CO2. And so BioCrow is built around what we call a multi-layer canopy model. And so it's a field scale model where we essentially are simulating a field of plants, and we split this field, the canopy of this crop or field, into different layers. And this allows us to capture the profile, the vertical profile of things like the sunlight through the canopy, as well as wind speed and relative humidity, which are then all inputs into this model. And so for each layer, then we have a portion of the canopy layer that is expected or simulated to be in sunlight versus in the shade. And we use those amounts to calculate the photosynthetic rate or the assimilation rate, along with things like the transpiration, which is the water that's essentially transpired from the plants into the atmosphere, and the leaf temperature climate for each specific layer. All these layers then get integrated together. You get a full canopy photosynthesis rate or assimilation along with evapotranspiration. This information then gets split into or allocated to different parts of the growth of the plants related to leaf, stem, root. And in this case, I've been working with soybeans, so talking about pod and seed. And then this gets updated. So the more leaf you grow, the larger canopy you're getting. And so you're updating now your vertical profile related to this. And then, of course, also soil water is an important component of this and impacts things like the stomatal conductance, which impacts things like evapotranspiration, as well as leaf growth. Okay. And so BioCrow, I worked with a soybean version of it, parameterizing and developing the soybean version. It was initially developed for other bioenergy crops, including miscanthus, I think was the first one. And so you have this framework that you can use for a bunch of different crops. You need to parameterize it specifically for the different crops. But you have also, so those are one set of inputs shown here. You also have your weather inputs, which you simulate at an hourly time scale of the solar radiation, temperature, wind speed, humidity, precipitation, And then you get a whole range of different outputs related to the biomass over the growing season or the growing period that you're simulating, leaf area index, which is another measure of kind of the size of the canopy, among other components. And so using this model, we calibrated it and kind of validated it against some data from Illinois at the soy face facility, and shown here. And so this is a field where they can control essentially the atmospheric CO2 levels for certain parts. If you can see, there's this kind of ring down here. And so inside of this, they're able to increase the atmospheric CO2 locally so they can grow crops in a field condition, but under higher levels of atmospheric CO2 and than what is ambient right now. And so we use some of this biomass data that I collected a few years ago at this point from some of these crops for looking at leaf, stem, and pod biomasses over 4 different years. And so we calibrated the model under ambient conditions on 2 of the years shown here, and then tested the biomass predictions under elevated for those 2 years without recalibrating the model, and also under ambient and elevated for the other 2 years that weren't trained on the model. And we were able to, most of the calibration was related to essentially how carbon was allocated in the model. And so once we have that, we have the model now, but the question is, not just building the model, but using the model for something. Our question is, how can we exchange things, or how will changing parts of the system, the crop growth, impact yield conditions? And so photosynthesis, if you go back to this, is kind of the key driver for soybean growth and yield. There are other components that can also influence, of course, but photosynthesis is one of the key components. That's how we're turning the solar energy into chemical energy in the plant and leading to the growth of the plant and the yield of the plant at the end. And so, it's shown here, the photosynthesis and the canopy assimilation parts. And so improving this efficiency of the photosynthesis is one potential way of increasing the crop biomass and yields. And so the thing about photosynthesis that as ECE majors you may not have heard before, it's actually really inefficient. If you think of the amount of energy is coming from the sun, the amount that is actually converted by plants into chemical energy is about 5% of that, and that's actually kind of at the high end. And so it's usually more around like 3%, but about 1 to 5%. And so there's a lot of different reasons for that, places where energy is kind of lost in the system. But the idea is that if we can increase this efficiency such that it's a little bit better, and it's a comparison with the solar panels that you could get to put on the roof of your house has about a 20% efficiency. I just kind of put that in a range that may be more familiar. To address, there's a couple different areas, or just increasing the rate of some of the photosynthetic parameters. And so that is the first thing that we started to look at with our model. And so photosynthesis occurs in the chloroplast of mesophyll cells in most plants, but not all, but in soybean this is true. Where this is a cross-section of a leaf, we have our mesophyll cells shown here, and then inside you see these green things. This is the chloroplast, and so this is where photosynthesis is occurring. And then inside the chloroplast, there's 2 main sets of reactions of the photosynthesis, the light-dependent reactions and the light-independent reactions. And the way that we model these, then, the most common model, I'd say, is what's called the Farquhar-von Camera-Barry model. And this is a simplified version of, so this is a similar metabolic pathway as I showed with the lignin model, but now looking at photosynthesis. But this Farquhar model is a simplified model, so it's not including every single reaction like we did in the lignin model, and really simplifies it into 2 main reactions shown here. And so one that represents more of the light-dependent reactions, sometimes called the electron transport rate, and then the other equation, which is related to this Robisco, which is an enzyme, carboxylation of the CO2, or the carbon fixation. And so I have these 2 rate equations describing this pathway, and then essentially whichever rate is limiting is what is calculated to be the photosynthesis rate, or the photosynthetic rate. Two key parameters in these rates there are what I've circled are what we call the VC max and this J or J max parameter, which are essentially the maximum rates that these two reactions can be. And there have been lab experiments that have looked at increasing these VC max and J max rates by increasing either the amount or activity of certain enzymes. And so in tobacco, this has been done where they increased the rates by about 10 to 30% and saw about 10 to 20% increase in the photosynthetic rates. And in soybean, experimented about 4 to 8% and saw a 4 to 14% increase in photosynthetic rates. And so these have been mostly in the lab so far, but our question is if we want to see, one, if we can get kind of similar changes with the tobacco and soybean in our model, and look at what we would expect to see happen in the field if we make these changes. And so here we did a sensitivity analysis of the VC max and J max parameters in our model, looking at our control or wild type shown here in the center, and increasing and decreasing both to plus and minus 50% of the control values of those parameters. This shows it under atmospheric CO2 levels of about 400 parts per million. I think today we're around 415. The summer it got up to around 420, so it's a little bit lower, but approximately where we are at ambient today. And we see as we increase these two rates, we do see an increase in our pod biomass, which is related to the yield, shown here. And as we decrease them, we see also a decrease in these. If we now look at, this is the plot I showed before up here, look at these now under different CO2 conditions, and I'll show a couple other parameters. And so we have, this is the pod, this is the shoot, so the entire above ground biomass, leaf area index, which is related to the canopy size, and then our photosynthetic, the daily maximum or the daily average over the growing season. And just as a note, so we only looked at, we increased the CO2 levels, didn't change the climate, other like the temperature climate along with those CO2, even though if you go into the future, all that's going to change as well. But what we see here, increasing CO2 levels already just kind of on their own, you do see this increase in assimilation and the pod and shoot biomasses. At the lower levels, you see more of how increasing the VC max and J max can also improve things. This is a little bit easier to see on this plot, where we're now looking at, this was kind of the absolute abundances that we were seeing for these values, and now we're looking at the relative changes. As you can more clearly see here, when we get to high levels of CO2, the kind of effectiveness of changing the Jmax and VC max are a little bit less, especially VC max. Most so see improvements in the yields and the assimilation by increasing the Jmax, which is related to those light-dependent reactions, part of the photosynthesis. And that's shown by, this line is showing kind of the path of steepest increase through gradient descent. Okay. And so these plots that I just showed are the average of about 10 years of climate data from central Illinois. But the intra-annual variation between these has some kind of surprising results of how well altering these parameters, these photosynthetic parameters, impacted or increased the yield. And so when we looked at each year individually, we see that particularly for two of the years, 2007, 2014, we actually see that when we were trying to increase the photosynthesis, we ended up with less pod in the cases versus the control where we weren't changing the photosynthesis parameters, which surprised us a little bit. And so we also looked, if you look at this LAI, which was the canopy size, or related to that, you can see it has a slightly different profile or pattern. We don't see those necessarily the same changes for the 2007-2014, but they were higher in those years than some of the others. So we thought maybe it's related to the canopy, but there's not enough information just looking at these 10 years to really make a strong conclusion from that. Because there could be there are other factors that also influence things. And so what we did next was essentially create a set of synthetic weather data to create 1,000 samples taken from using a bootstrap method of these 10 years of weather data to create a range of profiles of weather data that comes from weather that we've actually seen in this period. And then we ran for all of these 1,000 climate inputs, our growing season years that we're simulating, ran our model simulations, and looked at, so in this case, it's looking at the frequency, the range of variation of how much the pod increased when we were increasing our photosynthetic rates. And so we see quite a range. And so we took the upper quartile and the lower quartile and just compared those results to see if we could try to pick out what was happening really in these low cases where we weren't seeing an improvement or maybe even a negative improvement or not improvement at all. And so looking at the upper quartile, which is gray, and just compared to the lower quartile, you can see here the large range between the as predicted pods at the end. And again, looking at this leaf area index, or the canopy size, we do see a pretty strong significant difference between these two leaf area indexes between the two sets of data, where especially early on in the growing season, the set that ends up with a lower pod has a much larger canopy size than the canopy of the upper quartile that had the most pod. And we see this, if we just do kind of a linear correlation, we do see a trend between the size of the LAI or the canopy and the pod, negative correlation. And so this suggests that you can get, if you get a lot of kind of sunlight in the early part of the growing season and you have your increased photosynthetic rates, so you're creating a lot of biomass, most of that's going to the leaves at that point. Some of that will be remobilized later to the pods and seeds, but not all of it. But you've now had this larger canopy, and depending on the sunlight you're getting later in the season, when the pods and seeds are being developed in the plant, you could have a lot more shading with the larger canopy size, which leads to the lower canopy scale photosynthetic rates. And so looking at the how things change at different times in the growing season and the relationships, we found some correlations between particularly sunlight and temperature, as well as some as with humidity, influence how well these increasing or improving this photosynthetic rates will lead to actual increases in the yield. And so just some key points from this part of the talk: as CO2 levels rising, increasing the electron transport rate is going to have a much larger impact on increasing soybean photosynthesis, and such to the point that you could even decrease a little bit the VC max parameter, reallocate some of those resources to elsewhere without seeing a negative impact on the photosynthetic rates. Seasonal variations also play a significant role in how effective increasing these photosynthetic rates are at impacting the yields. And it's kind of suggests that in future climate scenarios, we should really be looking also at coupling increased photosynthetic parameters with other improvements such as to canopy architecture, which would impact essentially how much light is able to get through the canopy. So that brings me to the end. Just some quick acknowledgments. The lignin work, so the first project I talked about, was done here, and along with Jack and colleagues in the Forest Biotechnology group. The second project was done at UIUC, or in Illinois, as part of the Crops in Silico project and the RIPE projects, which are just related to looking at increasing photosynthetic efficiency. And the second project, most of this work was done by my postdoc, Dr. Yufeng He. Any questions?
I do a lot of service in food banks, and based on what I discussed, and I did not work in your field, so this is based on ground experience. We are able to, there's a lot of work into increasing the yield of staple foods, like soybean, wheat, but people do have inaccessibility to the entire nutrition palette, increasing the yield of vegetables and fruits and stuff like that, because those are some considered more expensive research. Staple food is somewhat less very expensive research, which many people can do. So are there any efforts to ensure that a person that has access to the entire nutrition palette? This is based on my experience in working in food banks. I do not work in your field, so my second question is very tangential to what you're presenting here. The word targeted gene research triggers a lot of weird memories of reading hours and hours of reading about conspiracy theories. So this is a very ruthless question to you. How effective are the national and supranational legislatures effective in helping you handle the ethical and moral ambiguities in targeted gene research? Thank you.
Yeah, so good questions. So first one related to nutrition. Yeah, so a lot of this work has been on the staple food crops. Part of that reason is you said it's cheaper. It's also easier. Where we are with kind of these engineering crops, plants are all very different from each other in some respects when you're starting to look at the engineering levels. So a lot of it's related to the data that we can easily get, and also how easy it is to make some of these changes at the gene modification level. And so even things like soybean, they're just now really starting to be able to make these changes in soybean, which is a lot of why previously I talked about some tobacco that's considered a model crop, because people were able to make these changes to tobacco a lot easier, even though that's not really a crop that we care about a whole lot from a food perspective. But the idea is that hopefully you test some things in tobacco and it would translate to the crops we care about. Related to that question, I know I haven't quite gotten to what your question was about yet. Related to that, there has been some evidence that shows, increasing yields can sometimes decrease nutritional content from what's actually coming from the soybean seeds that you get. And so there is also work going on towards, okay, we want to increase yields, but we also want to maintain this nutritional content. And so how can we do that? Or even potentially looking at if you want to have specific profiles of amino acids in soybean seeds, can we do that? So there is work, people looking at addressing that and working on that side of things. A lot of specifically the Right Project is interested in getting technologies in this kind of gene modification crops hands of small stakeholder farms, specifically in Africa. And so kind of giving the power back to these smaller, not so much the big corporations, but the smaller farms where people are using to essentially feed their families. Related to that, but I don't know, at least on this project, which doesn't mean that other people aren't working on it, the side of making sure that the full nutritional profile is made. I'm not aware of that. But there are other groups who do look at some of this research in other types, like fruit crops and everything, but it's a little bit more difficult because there's less data. And my understanding is that the ability to make some of these changes into the crops is not as well understood yet. Okay, second question. Right, the legislative side of this, GMOs, right, can be very contentious, and you raise the potential ethical questions around this. And so my answer on how well the legislative bodies do, it kind of depends. And it kind of depends on where you fall on the side of things. So a lot of the breeding that has been done historically is also looking at making changes to the gene, but from plants versus what we consider more of these gene engineering or gene modifications going in and targeting specific things. And so different countries kind of have different levels of regulation or comfort with this technology. I am supportive of it. I don't have a problem. So that's kind of my personal side of things, but there are regulations in place. And I know my colleagues who do a lot of these field experiments, you do have to be very careful when you're doing them that you're not kind of having the spread of these transgenes in your experimental plots to other plots and everything. I don't know if that quite answers your question, but yeah. Cool. Yeah.
I can say something about GMOs that
Yeah.
a lot of people don't know, but pretty much every type of wheat that you eat today was created by irradiating seeds to introduce random mutations such that it produced a phenotype. This is something that you can see with no regard to what other things were changing that you couldn't see. Whereas the kind of gene editing that we're doing now, I don't know whether it's good or bad, is targeted to a gene,
Yeah.
and they can actually track the off-target effects, which are the things that change in response to those genes. So it's just
Yeah. Thank you for adding that. Yeah. Yeah.
So in the first half of your talk, you mentioned that for phenotype predictions, you ended up going with the random forest model to treat it more as a black box. I was wondering, are there attempts to like try and reverse engineer what the random forest is doing, or ways of trying to figure out how the black box is working, rather than just being a biologist and doing experiments?
So we use another, not quite to the extent that maybe we should. We did, so one of the other kind of benefits of using the random forest and why I went with that approach is because it does give some measure of variable importance as well and variable selection techniques. And so you do get a little bit of information about what potential of the metabolites are maybe more important for specific phenotypes versus ever, but especially with some of those things like the wood density, there's a lot of other things other than the lignin pathway that go into wood density that's not really included in our model. So it's, I say that's something definitely to look into a little bit more, but we haven't yet, yeah.
So for the parameters for your models, are they fit? I think you were talking about Vmax and then J.
Yeah.
Mm-hmm. But in general, do you have to have values for those? Are they fit using?
So for the soybean model, most of those parameters, so the VC max and Jmax and other similar types of parameters, had been experimentally measured along with that biomass data, and so we use those measured values. What we didn't have that we had to kind of calibrate and do an optimization for was how carbon is allocated. And I'll say this is not my favorite part of the model, and one that I have a student who I'm hoping we're working on trying to improve it. But most of these crop models all work this way, where essentially carbon is allocated at like a certain growth stage or development. You say 15% of carbon is going to the leaf, 30% is going to the root, or so on and so forth, and this changes as you go through. We did in our model, we have, instead of being quite just what they call like a partitioning table, which is like a linear piecewise, we did a little more, we had kind of a exponential or logarithmic equation that we used, and we fit some parameters on that to get essentially, but it's still a partitioning of saying a certain percent of going at a certain time. So that was what we had to fit to the biomass data, but pretty much all the other parameters were collected from literature or experimental values. Yeah. Gotcha.
Great talk, Megan. So, photosynthesis, as you know, after 500 million years of evolution, is still unbelievably inefficient.
Yeah.
With some of the things you saw. And Dr. Donald Orr from your University of Illinois recently published a paper by simply combining genes from different species, he was able to substantially mitigate the photorespiratory effects and thereby increasing photosynthetic efficiency by quite a lot. So from an engineering or a modeling perspective, by understanding the kinetics of the enzymes involved and how the pathway works, could you in any way model or optimize, say, like the perfect photosynthetic pathway to remove most or all of the inefficiencies under different environmental conditions? Do you think that's at all possible?
Maybe. So maybe kind of two different answers to this question. One is there is actually, and I recently heard a talk from someone, people who are working on what they call, I guess, artificial photosynthesis or synthetic photosynthesis, where it's like you don't really even start with what plants do, but you just say is like, this is what I wanted to do, take the minimal amounts and start from there, which in some respects may be a little bit better, but that's not like what's happening in plants. The answer to your question, so I think maybe eventually we could, but it becomes a little bit difficult. One, when you're looking at under different environmental conditions and how that's going to change things. And also, so I know specifically with like the photorespiratory system, there is debate over, some of those outputs may actually be important for other processes in the plant. And so if you completely get rid of that, are you changing something else outside of photosynthesis that would negatively impact the plant or not? And so just looking at, I think you would need to have an even broader understanding of how making changes there would impact other parts of the plant as well. But yeah. But I have some work, we do have a model more like the lignin model on that scale of photosynthesis that I didn't develop, but the lab I did my postdoc in, some previous students there had developed this, and that we're trying to do some work with that more on this, not just looking at these two rates, but at all of the more individual rates making changes. Yeah. Yeah.
So, thank you for the talk.
Yeah.
Insightful. I'm actually not from engineering background,
Yeah.
but I'm interested in, like, you mentioned every model you built, I saw there, like, experimental data to support or at least, like, evaluate if the model is good or not. I'm just wondering, like, how many, like, do you have a screening process beforehand? Kind of just, like, sort out the model that is more important to be worth for experiment, because it takes a lot of effort and, like,
You mean it's like from the model predictions, kind of screening those to before going into the field, or is that what you're asking?
yeah, so like does every model need an experiment to support it?
At least initially, like, you do need to have some type of experimental validation you can do stuff without it, but there's always going to be questions of how accurate your model is under different conditions. And so having a validation, especially an experimental validation, is important to at least say, like, yes, this model is predicting things and that's what we see experimentally. And so we can trust it, at least under these conditions type of thing. Yeah. Yeah.
So when you have for example, you have the model of the soybean, are you expecting it can be translated with a few modifications to other crops, or you do like?
Yeah. So that's actually a really good question. And I think one of kind of a unsolved area in this field is, so as I kind of mentioned before, crops, like plants, work very differently from each other's. And so if you're trying to parameterize every model you do for every type of plant, like that's not really feasible. And so, but there's not really to date been a good way of systematically like translating from one crop to another without doing some reparameterization. But that is, hopefully in the future we'll get there. Yeah. Oh, yeah.
So is there any scope for a platform similar to the all-on-chip, let's say applying on chip, just to analyze the effect of this photosynthesis?
Ooh. I have no idea. Do you know of anything?
There are different programs like BioBricks, for example, that attempt to have synthetic pieces of biology or biological systems that you can try to put together. But does that translate to actual organisms that are actually out in the field? It still depends. So that's very much an open area. But it would be extremely beneficial if you had something like a system on a chip that could be systematically modified and reprogrammed. Yes, it could make significant advances in this field.
Yes,
Bye.